A very good afternoon, Doctor. We belong to Group 5. We are here to talk about a sustainable solutions to plastic waste management using Rhodococcus species. This is a list of my group members, Ho Yun Long, Tan Jingmei, Li Yuejia, and me, myself, Yi Mei Jin. So, first of all, plastic is a high molecular weight hydrocarbon polymer made from petrochemicals. The basic material used for making plastics are extracted from oil, coal, and also natural gases. Urbanization rapidly increases plastics product consumptions due to its versatile material, making plastic pollution one of the most important contemporary environmental issues. It is waterproof, resistant, flexible, and relatively cheap to manufacture, making it widely available across the world. So, extensive exploitations, poor recycling, limited repetitive use, and also plastic remarkable resilience to environmental and microbiological action cause massive garbage accumulations in land and also marine settings, endangering the human and also animal life. The persistent ability of plastic in an environmental scenario is causing pollutions. It is estimated that 30 to 40% of plastics are produced worldwide for different packaging purposes, directly rising plastic garbage at the rate of 12% per annum. In 60 years, worldwide plastic output has increased 24 to 300 million tons. The country China, United States, Germany, and also Brazil have major contributions in dumping of plastic in the sea as compared to other countries. Microplastics, partially degraded plastics debris of less than 5 mm in diameter have even shown to pose an even more serious impact on ecosystem by concentrating persistent organic pollutants. These are often hydrophobic compounds with a high affinity to microplastics, thereby entering the food chains when microplastics are ingested by wildlife. So plastic waste has become a global issue, and the need for sustainable solutions is more important than ever. Researchers have discovered that certain strains of Rhodococcus species produce enzyme known as PATase and also MHETase that specifically target and also break down the molecule structure of PET. Some of the advantage, such as the biodegradations, the versatility, the energy recovery, environmental friendly, cost effectiveness, and also potential scalability. Our research aims and also objectives include in plastic degradations to develop an efficient and sustainable method for plastic waste management, to optimize the degradation process to ensure maximum breakdown of plastic materials. For environmental impact, we want to prevent plastic accumulations in landfills and also the natural environment, reducing pollutions and also potential harm to wildlife. For process optimizations, to study the factors that influence the degradation process, such as temperature, pH, and also substrate concentrations, and develop strategies to enhance the degradation efficiency. So I will pass the next slides to my next team members. Thank you.
Thank you, Meishin. The methodology of Plastinator is divided into three phases, which are Phase 1, Research, Phase 2, Production, and Phase 3, Regulatory. My name is Yuan Long, and I will be explaining on the methodology in the research phase. Step 1. Bacterial culture and growth. Cryopreserved Rhodococcus ruber, strain C208 cultures were defrosted on ice, resuspended with sterile milky water and strict on nutrient agar plates, and cultured aerobically at 27 degrees Celsius for 4 days. Fresh colonies were picked from the plate and subcultured in triplicates in 100 milliliters of liquid nutrient broth and incubated at 28 degrees Celsius in Erlenmeyer flask of 250 milliliters on a rotary shaker at 100 RPM for 2 days. The mid-exponential phase of the culture was approximated based on the doubling time of Rhodococcus ruber and confirmed by photospectrometer measurements of optical density at 660 nanometers. Mid-exponential phase bacterial cells were subsequently centrifuged for 10 minutes at 1500 RPM. Step 2, which is upscaling. The stock-cultured bacteria were transferred into 5 liters Erlenmeyer flasks with the appropriate amount of liquid nutrient broth of the same ratio as the previous subculture and incubated at 28 degrees Celsius for 3 days. This provides enough bacteria for subsequent steps during research phase. Next, step 3, which is microencapsulation. This ensures that the bacteria survive and continue to degrade the plastic waste. Rhodococcus bacteria are encapsulated into microcapsules, which controls the release of these ingredients in a targeted manner. Therefore, the functionality and efficacy can be improved. Step 4, which is the efficiency assessment in simulated environment. In the first tank for natural attenuation, no special treatment was performed. In the second tank, for control, contained Rhodococcus rubra bacteria without microencapsulation, while the third tank for bioaugmentation contained microencapsulated Rhodococcus rubra bacteria. The samples were taken at specific intervals of 0, 3, and 7 days to analyze the efficiency of biodegradation by Rhodococcus rubra bacteria. In step 5, statistical analysis, since the experiments were conducted in triplicate and the values obtained were reported as the mean plus minus standard deviation of triplicates. Analysis of variance or ANOVA was used for statistical analysis using SPSS software version 22.0. Duncan's test for multiple comparisons was applied to express significant differences between the means at 95% confidence interval. I will now pass on the presentation to my colleague Meg for phase 2 and phase 3 of the methodology.
The second phase is the product development. First step, we will source out OEM, original equipment manufacturer from overseas, which could produce both bacteria, Rhodococcus rubra, and the soluble algae biofilm for our soluble pod. Next, once we find the suitable OEM in overseas, we will discuss our product's requirements with them. So for the bacteria Rhodococcus rubra, we will discuss in terms of formats, such as in freeze-dried form, and also application purpose, which is to degrade the plastics. Next, we will discuss for our concentrations, purity, and additional features such as scents and color. For soluble pod, we will discuss in terms of the specifications such as the dimension, thickness, weight, color, packaging design, and more. The third step is production and packaging. Once all the requirements are being discussed and confirmed, the manufacturers will start to produce plastinated products. After that, they will ship back the plastinated products back to Malaysia by flight. Besides the plastinator being shipped back to Malaysia, there are also some documents will be attached together with the product, such as the product information, product specification or certificate of analysis, material safety data sheet, and also halal statement. All of these documents will be examined by the Custom Malaysia in international airport for safety purpose. The last phase is the regulatory compliances and market launch. Since some of the standards in overseas is not as same as the standards in local, so we will send our plastinated product to local laboratory test and product inspection. We will test it under Chemical, Polymer and Composite section, CPCT, in Siring QAS International Sendirian Berhad, which is recognized by the Standards Malaysia number 087. Next, we will proceed to regulatory registration. First, we will register under the Siring Eco-Labeling Scheme. So this will make our product to be more competitive over the similar product in the market that prioritize the environmental considerations. Next, we also will register My Hijau mark for our product, which is due to the benefits of help our company to get tax deduction in the future. The third step is the plastinator soft launch. We conduct a soft launch event and provide free plastinator product samples to the invited audiences such as influencers, medias, and targeted customers. Next, we'll present our product features to them. Soft launch events is not only introducing our product features to the audience, but also will enhance the networking and relationship building, which can help us to find the potential partnership on investors in the future. The last step is the full-scale launch, which means that our plastinator will be officially launched in the market. First, we will make some marketing campaign to promote our product through different channels such as social media and influencers partnership. And our product will be distributed through retailer shops and our own online shop. Lastly, we will also make some sales and promotions such as special offers and loyalty programs to the consumers who bought our product.
Thank you, Cheng Ming. So now I'm going to talk about the expected results of our study. Through the study of various factors like temperature, pH, and substrate concentration, the project aims to optimize the degradation process. Hence, the first expected result would be the establishment of optimal conditions that maximize the breakdown of plastic materials and even microplastics by the bacteria. Secondly, the study should also prove the effectiveness of the microencapsulation packaging technique in protecting and delivering the bacteria for targeted plastic degradation. For instance, the controlled release mechanism of the microencapsulation packaging. This involves investigating the release kinetics of the encapsulated bacteria when exposed to target plastic materials. The release rate and duration can be measured to ensure that the bacteria are delivered in a controlled manner. Thirdly, the bacteria is also expected to be feasible in terms of culture and growth, as well as cost-effective when upscaling the production of the bacteria in mass. Next, I'm going to talk about the further impact of the study. The study of our product can contribute to scientific knowledge by providing insights into the plastic degradation capabilities of Rhodococcus species bacteria. It may lead to further research on the mechanisms of plastic degradation, the optimization of bacterial strains, and the development of novel enzymes or genetic modifications to enhance degradation efficiency. Furthermore, our product can also provide significant environmental and societal benefits. It can help reduce the environmental impact of plastic waste, minimize pollution, protect ecosystems and wildlife, and improve public health. It can also contribute to sustainable development goals, promote responsible consumption and production, and foster a more environmentally conscious activity. Also, the study's results could have practical applications in various industries such as the waste management, recycling, and biotechnology. It could inspire the development of the new technology or processes that utilizes the Rhodococcus species bacteria or similar organisms for a large-scale plastic degradation, leading to the creation of innovative and effective technologies for plastic waste treatment and recycling. Last but not least, the findings of the study provide evidence of a sustainable approach to manage plastic waste, which can be considered by waste management authorities or either the policymakers. The study may influence the development of regulations and guidelines that promote the use of microbial-based solutions in the plastic waste management. These are the references used for our presentation. So basically, that's all from our group. Thank you.